Good morning, everyone. Thanks for coming today. It's my pleasure to introduce our first speaker for the 2023 IVB seminar series, Dr. Dane Wittrup. He is currently the CP Dubs Professor of Chemical Engineering and Biological Engineering at MIT. He's been responsible for many recent advances in the directed evolution of biopharmaceutical proteins, as well as the display of libraries of antibodies and other binding reagents on yeast, or commonly known as yeast surface display. Dr. Wittrup's research interests are primarily focused on the cancer immunotherapy using evolved proteins, especially cytokines and antibodies. He's also the co-founder and chairman of the SAB at Adimab. He's a member of the National Academy of Engineering, and he previously served as the associate director of the Koch Institute for Integrative Cancer Research at MIT. So please join me in welcoming Dr. Wittrup.
Thanks very much for that generous introduction. If you ever wonder if it's a good introduction, I think clap before you started speaking. It was a good introduction. So I'm going to tell you a story today about how we try and get immune stimulation to occur where we want it and not elsewhere. And so the title that I also gave that was publicized plays on the idea of Occam's razor being the simplest answer being the correct one. And Occam's Pharmacokinetics would say that if you want to get a drug into a tumor, that's your objective, maybe what you should do is put the drug in the tumor. But you're not necessarily done there. You also need to make it stay there, which is the final piece that I think we've found some interesting ways to do. So to give you a sense of what I'm going to talk about in the next hour, why would you do this? And I'll give you some motivation for it. How might you go about keeping these drugs in place? Does it work? Really well. Does it work in the places that you didn't inject? The answer is yes. And how does it work? More specifically, which parts of the immune system have we actually influenced this way? And can we learn something about how to design these molecules for this mode of administration? So let's start at the beginning. Why administer a drug directly into a tumor? So step back and think in terms of how cytokines, the signaling molecules that immune cells use to communicate with each other, are deployed by the immune system. It's not crazy to think you might learn something by seeing how they were used by our immune system, because our immune systems use them really effectively. Well, what they do is they spatio-temporally program their use. These cytokines are expressed in certain places at certain times for certain periods of time. And so take as an example, IL-2, it's a low-numbered cytokine. It's discovered early on. It's important as a T cell growth factor is how it was originally discovered. The way it's mostly deployed by the immune system is for very brief periods of time, hours to say half a day or so. There's a tight negative feedback loop on for how long it's expressed. Mostly in the lymphatics, mostly by helper T cells. There are exceptions to all these things. All biology is practically nothing but exceptions, but IL-2 is mostly something that's there for a short period of time in the lymphatics to expand T cell populations. IL-12, different. It's expressed actually also in the lymphatics to some extent, but more in inflamed tissue. And interestingly, it has more of a positive feedback loop in that expression of IL-12 drives expression in other cells of interferon gamma, which subsequently drives expression of IL-12. And in the absence of some other suppressive regulatory mechanism, it would spiral out of control forever. So these two, and not to put them on the spot, but Johnny asked me before the seminar, which do you like better, IL-2 or IL-12? And I have a slide for you, Johnny. Here we go. Apples and oranges. They're both very powerful at the particular things they do. But we, being human, think we know better. And so what we do instead is make a bunch and flood the person with it for as long as they can handle it. And that's better. That's what I'm going to do. But biomimicry is a really powerful principle because natural selection really gets rid of a lot of bad ideas. over the millennia that it occurs. And so for intratumoral immunotherapy, ITIT, these cytokines that the immune system uses are generally not endocrine signaling factors that go everywhere in your body. But that's how we dose them. We dose them intravenously, soak the person in this drug to the highest level they can tolerate, to the maximum tolerated dose. And that's not mimicking the natural exposure. Does that matter? Maybe not. Maybe we can do better than nature because we're trying to do something different. But might not be bad to think about trying to do the same thing. And from first principles, if you keep the drug where you want it, you won't be exposing unnecessarily the rest of the body to it. And also you'll be getting better area under curve on that tissue. So the history of clinical application of cytokines is one of frustration because you have a hard time getting enough of the drug, be it say IL-2, which is an approved drug, or others which have not yet become approved, into the tumor because you go through this process of soaking the whole patient in order to hit the target tissue. So if you look at the therapeutic window as the range of concentrations in which you're getting clinical efficacy without unacceptable toxicity, sometimes it's such a narrow window, it's actually negative, it doesn't exist. Sometimes there is no window at which you can increase the dose sufficiently to write up the green curve of efficacy without having already ridden too far up the red curve of toxicity. So trying to pull those two apart is what we are about and what a number of other approaches are about, which I'll give lip service to here. Other people are trying other ways to do the same thing with cytokines, trying to target them by fusing them to antibodies, has some issues, but there's active efforts in that direction. Trying to express them locally through transfection, trying to mask them in the periphery and then have them unmasked by proteases in the tissues. I'm not going to do the thing now of saying that these guys are all wrong and I'm the only one who's right. These are all legitimate things. I hope they all work. These are different approaches to the same thing that I'm going to tell you about. So just putting these cytokines into the tumors doesn't work because it leaks out too fast. And this has been tried, and I'll show you data on the next couple of slides, some of it locally generated. If you inject these cytokines into tumors, they leak out, and you end up kind of right back where we started. So you need some form of retention. This was some early work from my colleague, Daryl Irvin, where he was putting an anti-CD40 agonist antibody into mouse tumors. And it was fluorescently labeled here on the left. And after 2 days, it was all gone. After a question of just a few hours, it was already out in the blood and causing toxicity. So proteins as large as an antibody, 150 kilodaltons, leak out pretty quickly. Cytokines being smaller, more like maybe about 20 kilodaltons or so, leak out faster. And here was some locally generated data, which this nice paper from Professor Thomas, looking at different sized fluorescent tracers and looking at the 3-day area under curve for exposure. You see that once you get out of sort of the nanometer, the nanoparticle scale down towards protein scales, it's just vanishingly small exposures. This is also true in people. This is not just a mouse phenomenon. IL-12, after having a really unsuccessful IV dose clinical trial, people went back and tried to do intratumoral administration of IL-12. And what they found is if they injected it directly into a tumor, it appeared at peak in the plasma just a few hours later. And then looking at a subsequent pharmacodynamic marker, interferon gamma, that peaked about a day later. And to quote the paper, there was remarkable toxicity at essentially the same dose as was found to be the MTD for subcutaneous dosing. So this is a phrase you never want in your paper describing your drug. Remarkable toxicity. More recently, trying to express IL-12 using a gratuitous inducer in glioblastoma, what they found was, first of all, kind of a pretty haphazard dose response for the inducer. But this is looking at IL-12 in circulation after dosing, the expression was in the glioblastoma. So you need to do something to try and retain. So how do you do that? How do you try and keep them in the tumor? I'm going to show you two different strategies that we're pursuing and show you the people who did it. So first, the idea is to use collagen that's already there. Collagen is everywhere. It's at high levels. It sits there for a long time. And so it's a good target. Noor Momen developed this work and was the first one in my lab to do any intratumoral work. Published this in Science Translational Medicine 3 years ago now. Coming up on 4, I guess. And licensed it to Cullinan Oncology, which is developing an agent which is an IL-2, IL-12, albumin, collagen binding domain fusion. And they'll be filing their IND sometime this spring. Then, as we were doing that work, since my office sits right next to Daryl Irvin's, and we often, literally talk at the water cooler, which is right outside our office. It's the best office on campus. It's a good one to have to be sitting next to Daryl. We took a joint student, Yash Agrawal, who is now defended, but what we do there is we provide the anchor with the drug, rather than depending on the presence of collagen. We use a site-specific modification, a phosphorylation of a tail of a cytokine to preload it onto alum, which, because it's insoluble and it's in micron size, lumpy aggregates, stays where you put it. This technology has been licensed to a different company, Oncura, and they also will be in phase 1 trials, filing their IND in spring of this year. So part of the problem with any approach that has a diffusible cytokine is this kinetic competition between leakage and consumption. So there's a term of art, target-mediated drug disposition, TMDD, which is how fast your target cell takes up your drug. So the TMDD rate would be this agonism rate here with the T cell that sees the cytokine of interest. But then you've got a leakage rate. Now, if you were going to just inject the cytokine, if you put just the right amount in, conceivably, you could get consumption before too much of it leaked out. But the thing is, the number of T cells change, and tumors vary in terms of how much they leak. So getting that balance right in any sort of robust way would be difficult. You can try to moderate it by delivering as a depot. And when people think of drug delivery, this is the first thing they think of is some sort of polymeric material, which will entrap in an unavailable format the payload, which then is released. But then you've got that same problem. You have to match up consumption with leakage. So you want to consume it faster than it leaks. So pretty much the only things that we're interested in are things that don't go away on long time scales, anchored either by binding to the collagen or anchored by binding to alum. It's an important distinction because what I find when I talk to people about this, they often visualize this because they're about a mole Langer proteges who do this sort of thing. And there's order 1 people who do this sort of thing. So that's the difference between them. So this was a trick that we picked up from Daryl. He had published in Nature Medicine that if you take antigens and attach them to alum, not by physisorption, but by this site-specific phosphorylation. If you phosphorylate serines, threonines, or tyrosines, really, they bind almost with a covalent affinity. It's a very tight bond with this ligand exchange to the matrix. So what we end up doing here is co-expressing a site-specific kinase and are expressing this in stable CHO lines. It secretes a protein that then has these attachment points just on that one peptide, mix it with alum, and then administer. So where these two different approaches go differs. Collagen happens where collagen happens. So collagen 1 will be in the capsule, sort of the scar tissue surrounding a tumor. Collagen 4 is part of the basement membrane, and so it'll surround blood vessels. This staining was done with the protein lumican, which binds both collagen 1 and collagen 4. And so this is from Noor's STM paper. And you see it co-localized as well, both with collagen 1 and collagen 4. Conversely, alum, it just kind of smooshes out. When we inject it, it's in smaller flocks initially, so they spread out a bit. Over time, there are generally depots that form over the course of about a day. And bioavailability in that is probably low, I would think. So they're different. Which is better? Don't know. It's one of the things we want to find out over time. There are arguments in favor of either. So for example, in the excluded immune phenotype in tumors, often there are T cells running around in tracks in the collagen that surrounds the tumor. So that would be a good place to have IL-2 and IL-12. These two approaches also differ in their kinetics. So Nor did a very expensive and difficult experiment that I thought would be cheap and easy, which was quantitative PET. So I read and believed all the stories saying how great PET is at giving you numbers, absolute numbers of, picomoles and everything. Not that easy. There's a lot of massaging and correcting and phantoms and doing stuff. And Nor Moman spent like 3-4 months doing this stuff, and Nor months or people years. And so anyway, so this is what it looks like, and I'll even spin them for you. She made 3 different constructs. One that was a bivalent binder on the top fused to albumin, one that was monovalent, and one that had a dead version. This is Lair, not Lumican. It's a different binder. And there's some kind of cool phenomena here. One thing mostly is, of course, mostly the tumor is what's bright, even all the way out to 3 days. But you do get, the draining lymph node, then also downstream the axillary lymph node up there. Here, where there was only a single binding site, you get some liver uptake. And when it's dead and it was only size retaining it, you get more of that and sooner. And also you get more sort of fuzziness around the boundary of the tumor itself. But the time scale is roughly a day or so. So we get about a day or so of saturated cytokine effect from the collagen retention. By contrast, when we anchor on alum, it's there for a really long time. So here we were doing quantitative SPECT, and the company did this. This is Oncura data. And there you see a very tight tumor along the bottom, and you can see here that it reaches a point that about half the material, and this is labeled on the protein, not on the alum, is still there, all the way out to 21 days. When we look at interferon gamma as a PD marker for that, it starts to taper off by the end of 2 weeks, but we get about 2 weeks of exposure. Is that good? Kind of seems like it should be, but there could be such a thing as too much of a good thing. So again, we don't know for sure, but at least we have a tool to sample those different ranges, days versus weeks. Here's the contrast between what's in the tumor and what's outside the tumor with the alum anchoring. And so these are on log scales. So if you look at day 1, day 3, you can see that having anchored it on alum gives us about a 2 to 3 log improvement in retention in the tumor on the left panel. And then in terms of what's out in the blood, get about a 1 to 2 log improvement. And so we've shifted those curves in the way we wanted, both within the tumor and outside the tumor. So back to this graphic, there was less in the blood, there's more in the tumor. So we could dose up to a point that will maximize the tumor effect and haven't even begun really to see toxicity. Okay, so how well does it work? Works pretty well. So here I'm going to compare collagen to the alum for a B16 melanoma tumor where we're just administering IL-2 and IL-12, not just IL-2 and IL-12 plus anti-PD-1. And what you see is if we don't immobilize the cytokines, under half of the mice were cured, and when we do, more than half. The same Kaplan-Meier curves seem to be what we get with alum retention. So these don't look that different in terms of efficacy. Although in this case, we just did a single dose of the alum, and we did a couple doses over there. So in this model for these payloads, they look kind of similar. In terms of toxicity, they also look similar, just using body weight as a gross measure of toxicity. You give the exact same dose, but don't retain it. The mice come pretty close to euthanasia criterion, getting down to 15% body weight loss. But if you retain it, they really don't lose any weight. Same with the alum. So by that measure, they look about the same. Let's look at some more models, and I'll show you some more mouse data first. We tried a harder problem. In Nor's paper, we took a genetically engineered mouse model, which uses BRAF P10 mutations in a genetically engineered mouse model to induce the tumor. And we found that if we administered non-retained intratumoral cytokine, that's the blue curves there. And see, this experiment takes like 3 months to finish. This is an experiment that we hadn't finished it when we submitted the paper, finished it, and then when they came back, the reviewers didn't ask for it, so we put it in anyway. So like a bonus. It's a little thing you could do, by the way, if you really want to put the stuff in the paper, wait till later and just add it in. So when we dosed the blue curves where it could diffuse out, we cured over half the mice. That's the good news. The bad news is the ones that we didn't cure, we killed with the treatment. Okay, so about 1/3 to 40% of the mice died from the cytokine treatment. Whereas with the retained, we cured almost all the mice and none of them actually died from treatment-related toxicities. And the penetrance was perfect. Every one of the mice that you set up the tumors in, they all eventually die. Generalized a little bit further, this paper just came out this year, taking type 1 interferons and localizing them. We tried both collagen and alum. Worked better on alum with type 1 interferons than it did with collagen. I don't know why, but it did. And when we combined it with systemic IL-2, we got robust cures. We also worked with Richard Hines with an antibody that he had made, a camelid antibody against a splice variant of fibronectin, what he calls E3B and Dario calls EDB. And when we affinity matured it to picomolar affinity, and as you can tell, the 2 nanomolar probably worked just about as well, and intratumorally administered IL-2, we would cure over half of the mice, whereas if we systemically administered the same dose, we cured none of the mice and had barely any effect at all. Okay, so that's some mouse data. Now I'll show you some more mouse data, now trying to get at the question of what about the non-injected tumors? To what extent does a systemic immune response help us out here? Just looking at my clock and it said 38 minutes. I thought I have not been speaking for 30 minutes. We set it up early, sorry. So here we have to step back and be a little bit pedantic and say, okay, things have changed in oncology in the following way. For all time, up to about, say, 10, 20 years ago, the point of a pharmacologist who was trying to cure cancer was to kill every cancer cell. Your job was to kill every cancer cell. And if you didn't, that one would grow out and the patient would relapse. So that was the received wisdom. My job is I'm going to kill every cancer cell. If I don't, then I haven't done the job. That's not our job anymore because it's not possible. For one thing, there's too much genetic heterogeneity and spatial heterogeneity and spatial transport issues with the drugs. You're not going to kill every tumor cell ever. The only person who ever got cured from cancer was cured because there was an immune response on top of whatever the therapy being administered. So your job as a drug maker is not to kill every tumor cell, it is to vaccinate that individual against their own cancer. And so you're trying to develop a CD8 T cell response that responds to antigenic differences in that person's tumor. Well, now when we think about vaccines, we think about them very differently. We all got vaccinated against COVID. We all got vaccinated against COVID here. Is that because COVID enters our body here? No, of course, it's silly, right? But you're vaccinated in one place with a site of delivery of antigen and intense inflammation, which develops an immune response that protects you everywhere else. The immune system is constructed so as to go everywhere. So it does that part for you. So one site of intense self-vaccination is much superior. If you took vaccines intravenously, they probably wouldn't work very well, and you'd feel a lot sicker too. Okay, so how would that work in the context of a tumor? Well, the idea is if you generate cell death in an immunogenic context that's highly inflammatory, that antigen and danger signal will be delivered just through drainage, through the lymphatics to the lymphatic system, the lymph nodes that are local. T cells that respond to those antigens will be primed and amplified. They'll then leave those lymph nodes and go out into the periphery. They'll revisit the original tumor, but they'll also go to other sites. And so to do this job well, and perhaps also some of that job up there too, is enough because these guys will go everywhere else and finish the job. So here are some examples of that. We used a resectable 4T1 model where here you put this syngeneic breast cancer line in the mammary fat pad of the mouse, and it begins to metastasize, and it goes to the lungs and elsewhere. And so what we did was we dosed IL-12 into that transplanted tumor, and then also systemically gave anti-PD-1. After a couple of doses, resected the tumor that we'd injected. So we put a drug in there and then took it back away. Then looked to see if the mice survived or not. All the mice survived when we localized the IL-12. Almost none of the mice survived if we didn't. So having that local site of intense inflammation and plus checkpoint blockade actually gave a dramatically better response for metastases than allowing it to diffuse out, which you might have thought would have helped. Here's another example, again, from Noor's initial work. We did a 2-tumor model and a contralateral and an ipsilateral tumor. And you would inject IL-2 just into one of those two and either retain it or not. And then systemically, we added an anti-tumor antibody because those two work synergistically together, the antibody and the IL-2. And what we found is in the case where the IL-2 could diffuse out of the injected tumor, the contralateral tumor faster. So in the case where the cytokine could get over to that other side, it didn't work as well. So these are the treated side and the untreated side growth curves. And the untreated one grows out faster when you keep the IL-2 over on the opposite side of the mouse. So it actually works better to have one site that's more intense. Here's an example like that with alum and with MC38 tumors. And here we gave a single shot of the cytokine at the beginning of this experiment, which was not at the moment of inoculation of the tumor. The tumor had been in for, I think, 8 days. And only injected the cytokine on one side, but then gave PD-1 antibody elsewhere. And there's rational reasons to combine with PD-1, also practical reasons. Rational ones are that IL-12 leads to interferon gamma, which leads to PD-L1 upregulation, so it makes sense to try and block that axis. The practical one is most patients are going to be treated with anti-PD-1 antibody pretty soon, not already. And so we expected synergy and got it. All the mice were cured of both tumors from this. If it was the PD-1 only, it was resistant. Then we took those cured mice and rechallenged, and all of the cured mice resisted rechallenge with that same tumor. Okay. So how does it work? So a while back, Mikhail Tate published this nice paper showing this positive feedback loop between dendritic cells and T cells, where T cells secrete interferon gamma, which activates dendritic cells, which secrete IL-12, and back and forth. So an obvious thing to do would be to see if we needed interferon gamma for our efficacy. And we did, absolutely. If we add anti-interferon gamma antibody systemically, it's as if there's no therapy at all. The tumors grew out as if we had done nothing. And this is with alum IL-12 and anti-PD. Another sort of unexpected benefit was we got much better antigen presentation. And so this is an experiment where you take your tumor and get it to express a fluorescent protein so that if tumor detritus and debris gets taken up by a dendritic cell, you can see it as a proxy from the fluorescence of that fluorescent protein, ZS green. And so if we look 6 days after injection of alum IL-12 as a monotherapy, the fraction of cells that were positive for the green plus this signal to the CD86 is much higher. So the scatter plot looks, pretty convincing, but I really like the flow plot too, in that you can see here on this axis we have, the ZS green, so this is antigen. And there's just a much bigger population having taken up antigen than we would have expected. IL-12 alone, we didn't think would necessarily have that dramatic an effect, but it turns out there were literature precedents and high expression of CD86. So it's a priming machine. Just having administered alum IL-12, there's a whole lot of antigen being presented to naive T cells to prime new T cells. We did the usual thing of depleting a variety of different immune cell types We used a genetic depletion, essentially. These bad F3 knockout mice don't have the dendritic cells that cross-present. So on the far left panel, all the mice die. They don't have cross-presentation. We used CSF1R antibody to deplete tissue-resident macrophages. That's the light blue curve in the middle panel. And the mice die if they don't have tissue-resident macrophages. And we did CD8 T cell depletion, and again, the mice die. So these are the usual players, although the importance of the macrophages we hadn't expected, to be honest. But we expected the bad F3 to fail. We expected the CD8 depletion to fail because that's the magic. The moment of truth in an immunotherapy is a dendritic cell presenting antigen to a T cell. And so if we don't have one of those two, we didn't expect it to work. So how does it work in real cancer? Okay, real cancer. Real cancer doesn't just happen in people. Real cancer also happens in other vertebrates. So I was giving this seminar a few years ago at University of Illinois. I met Tim Phan, who runs a comparative oncology program there. And he serves as a catchment for people, Chicago, Indianapolis, St. Louis, who will bring their dogs who have cancer to Champaign-Urbana, and he will treat them with investigational compounds. And so we said, let's do this. And I said, let's do this. This is great. I went back to MIT, sat down with Nora. We're going to do this. This is great. She goes, how much protein do we need? That's a typical PI thing to do. Saving grace is, tumors not that big. Dogs can be pretty big. Tumors not that big. So the answer is we made most of the stuff we did for these trials in transient HEK cell transfections. We made cell lines to make more, but we're dosing, and also they're cytokines, so they're highly potent. So we're talking microgram dose. But the design cycle here for us is we've done a whole bunch of mouse work and have kind of reached a point of diminishing returns in terms of translation, understanding, what might happen. Now we're going to work not with a model. It's not a model of anything. It's cancer. These dogs have cancer, and see what happens. That is both inspirational and also will inform clinical trial design, because both of these companies are well aware of the things we're doing there. And Occam is now actually running their own alum trial with Tim Phan. And then in the course of this, we also turned around and rechecked things in mice, because we found, for example, there were some regulatory gene expressed, some suppressive regulatory genes expressed in the dogs. So we said, well, what if we block those? Let's check it in the mice. So we're going through design cycles relatively cheaply and fast compared to actually having to test things in humans. So I'm going to tell you about two ongoing experiments. The first one was just a biomarker study and safety study in sarcoma. Soft tissue sarcoma in dogs is when your dog has one of these. And these can be really pretty dramatic looking. And they're usually resectable. And then they usually locally recur. And so that gave us a lot of material to work from to do gene expression studies. It was not the right study to do for survival, but we're doing that with a melanoma study, which I'll show you next. So the protocol, it's a little small print here, but we gave a single shot and looked a couple days later. We gave, and looking means resecting, so it's just, we can't do any time variable there. We gave a single shot and looked a week later, or we gave 2 shots and looked a couple days after that second shot, or we did nothing. Now, the idea with the 2 shots is that the first one will give infiltration of immune cells. It will be there to respond to your second shot. So the idea was we would get a sort of a resonance that way. And so these were well tolerated. We had no adverse events in these dogs. The tumors, for the most part, soaked up the dose we were delivering. Looking out here to 2 days at the blood samples, and this is averaging over all the dogs. Interferon gamma did go up, but only about several fold over baseline. Got IL-10. So these are natural blood pharmacodynamic markers for IL-12 treatment. And then looking at the immunohistochemistry, it's kind of exciting. Quantitations over here on the right, and then example images on the left. When we did the double dosing and looked a couple days later, massive macrophage infiltration. T cells also go up significantly over the untreated. There's enough variation here that there was no significance for the earlier ones. You can see the brown dots in there, massive numbers of brown dots here. So then we did nanostring on these samples. And there's been an expert panel that developed a canine immuno-oncology set of probes for nanostring, which is great for us. So there's like 800 genes that it would probe. We probed those. And here what I'm showing you is, there's a lot of different ways to present this sort of data, but in the interest of a accessible presentation, I'm taking gene signatures with the titles above, which were found to be enriched in certain subsets. Then I'm showing you what the genes were within those gene signatures. And then each row here is a different dog. Okay, so you get a sense of variability because each row is a different dog. Far left, you have the 4 different protocols. Untreated is at the top. And so by and large, untreated, everything's blue, nothing's really happening. 2 days after a single dose, it's a whole lot of red running all the way across for all 3 of those dogs. And that corresponded to T cells, NK cells, macrophages, cytotoxicity, antigen processing. These are an output. Those signatures are an output, not an input. It was checking against all sorts of gene signatures, neurological development, infection and everything. But the ones that it spit out were these very basic immune response ones. Well, we were putting cytokines into a tumor. So it's what we'd hope, but it is what happened. If we look a week after the first dose, interestingly, everything's quieted back down. So it doesn't last that long. This was collagen binding. It wasn't the alum. So looking a week later, it was all blue again. But then if we redosed, again, everything went red. So we are successfully getting these tumors to get riled up, to get hot, per se. So how close is the analogy to what you might want to achieve in the clinic? Well, we did the same protocol in mice and use the mouse IO nanostring panel. And then we found this widely accepted gene expression signature in the clinic called the Tumor Inflammation Score. And it was first developed from looking at TCGA and also from some clinical trials. What they found is this tumor inflammation signature, which is these genes along the left, which are mostly interferon response genes, predicted reasonably well response to PD-1 blockade in humans. So if you look here now, we have the different protocols, untreated, one shot and wait, 2 shots. And in the mice, it's that whole column's pretty red here. In the dogs, C2 is pretty blue. So after a week, it had really damped down. In the mice, the first shot did some, but not that much. The second, waiting a week, it was still sort of inflamed. So that's a bit different. The suppression in the mice was not as complete as it had been in the dogs. That's encouraging that what we're doing in these soft tissue sarcomas in dogs looks like what you would want to do to get a response to anti-PD-1 in a person. Okay, so now for some therapy. So it wasn't done chronologically that way. We did them at the same time. But just written up and submitted the sarcoma paper, and this one is still reading out. So this was kind of a throw everything against the wall and see what sticks experiment because, these dogs were dying of their tumors and we sort of wanted to actually do something that would help them. And also we wanted it to work and then we could go about backing off and taking things out of it. So here's everything we threw into this. We gave a single dose of external beam radiation, which is the palliative care that these dogs receive because they're inoperable. By the time they get these tumors, it would just be too invasive to actually try and carve away all the surrounding tissue and bones. So we gave 1 shot of radiation with the intention of generating some antigen and inflammation so as to help prime a response. And then a day later, we started on a combination dose of IL-2 and IL-12 retained by collagen binding, all canine sequences. And then gave 6 of those cycles of treatment and then gave CAT scans on a monthly basis. And we tested this initially in healthy beagles under the shoulder in intradermal injections to find the maximum tolerated dose, and then did a dose escalation in these tumors. So here's what the waterfall plot looks like. So this is from CAT scan data. Along the bottom is the tumor stage by their classification system, and the color coding on the shading is from our dose level. 5 wasn't well tolerated. Those dogs did get sick. We had to back down. 3.3 was well tolerated. 2 is very well tolerated. And what you see here is that at dose level 1, 2 of the 3 worst responders were over there. But at those higher doses, we're getting fairly dramatic responses. This is a really good waterfall plot. So in addition, and now the number is actually up to about 80 doses, there was only 1 grade 3 immune-related adverse event, and that was liver enzymes were elevated, which happens in immune therapy. Dog was asymptomatic, there's nothing wrong with the dog, and it self-resolved after a couple of days. So now I'm going to show you some pictures of tumors in dogs. Just fair warning. It's kind of gross looking. They're not the grossest tumors we've had. Some of them are really horrible. You know, before you work in this field, you think of a tumor as a spheroid, or if you're really creative, maybe it's an oblate. God awful. These, different colors, different shapes, separating horrible, nasty things. These are not going to be too bad. So I'm going to show you 3 exemplary responses. We don't usually see the whole dog, but we always see their muzzle, and so they always have gray whiskers because these are all like 12 to 14-year-old dogs. And their owners drive 2 to 3 hours, treat the dog, they'd spend the night, and then take it back, and they do that 6 times. So we just feel good that things like this would happen, where just completely cleared that one. This tumor had subsumed that tooth entirely, and it's just gone. And we're now out at 1.5 years. That dog's running around in their backyard and so on. And there was interesting things that happened around day, what was it, like 130, when they came in for their monthly checkup. There was a new lesion here. Like, oh crap. So it goes. Went home, came back a month later, it was gone. So we're getting the equilibrium and elimination going on in real time. So that's Max Martin. Here's Maverick Miller, who had 2 different tumors. We injected them both. And gray whiskers on the muzzle there. And now we're coming up on 1 year, and it's just a tattoo because these are melanoma. So there's melanin, the pigment that persists even when the tumor's gone. Desi Ostrander had a really gross one. That's the tip of the iceberg. About twice that mass was back up in the cavity behind the eye. You can see some of it was pink, some of it was black. But that all cleared. So 1.5 years, that one's entirely gone. So pretty exciting. Like I said, it's inspirational towards what we're going to be doing in the clinic, but also gives us a more rapid turnaround for a design cycle if things don't go the way we expect. Like one of the things we would like to add to this would be checkpoint blockade. But to make anti-PD-1 antibody for these dogs, that would cost about $5,000 per dog for us. We did look into it. And I was full speed ahead, but that, hit that wall. It's like, I don't think we're going to do that. But it turns out that Merck Animal Health has regulatory approval to market an anti-PD-1. They say they're going to start marketing it in 2023. When they do, we'll buy it, use it to see how it combines. Because in the mice, as I mentioned a while ago, anti-PD-1 combines with these cytokine therapies in a rationally synergistic way. Okay. In the last couple minutes, I'll give you some real engineering here. Here's some numbers and models. So how big should we make these things? Had that conversation actually this morning. For retention, does size matter? How tightly does it need to bind to what it's binding to in order to be retained? So what Normoman did was write a super reductionist PK model here, compartmental model. I'm accounting for only the processes shown here, binding to an anchor, the anchor going away, being consumed by the cells, clearing. As it turns out, on the time scale we cared about, the clearance of the collagen was irrelevant. And so we found that with the mice, at least, with their teeny tiny little tumors, we were getting a lot of fluid spillover because it was just not that expandable as we were injecting only like, 25 microliters, but if you went up to like 50, a whole bunch of fluid came out. Not out of the mouse, but out into the mouse. And so we're mostly just capturing this balance between this kinetic competition between getting taken up or being released. So this is what schematically it looks like. These are those constructs that I showed you the spinning mice PET scan figures for. And the color coding is the model prediction. So the way we decided to use the modeling was we said, doesn't matter if you're at 100 times the EC50 of the cytokine or 10 times or 1,000 times. No, it doesn't matter. So what we plot here is the time at which the cytokine receptor or its effect is saturated. So the color coding, the yellow, aqua, purple, is days of active immunotherapy. And the yellow is only one full day, as I said, doesn't last forever. So when we had the bivalent collagen binder, we're predicted to get a full day of effect. One of the two dead, still predicted to get a full day of effect. Both of them dead. This is big, so it diffuses out slowly. So we get maybe half a day of effect. If we just put the monovalent binder, which is affinity-wise the same as this is the size effect. Going from this to this, you go from aqua to yellow. So being bigger definitely gives you more of a retention effect. And pure size for the small thing is pretty lame. Doesn't bind collagen at all. It's small. So it's predicted to be sort of down around the negligible. So this is all just PK. It's not effect. But it predicts, and it was literally predictive. We did all these experiments and modeling. Then we did the therapy trial. So prediction here is, number 1 and 2 will be best. 3 and 4 should be about the same, and 5 should be worse. So that's the way it worked out. So she combined these agents with the anti-tumor antibody. 1 and 2 cured majority of the mice. And they're the ones that had, say, a full day of IL-2 effect. 3 and 4 delayed outgrowth, but cured maybe only one mouse out of 10, say. And 3 and 4, they're both sort of in that aqua-ish range. 5 didn't cure any of the mice and barely delayed it at all. So that's just being injected but not being very big. So actually even the effective size alone going from 5 to 3, those are both non-binders. Or for the binders going from, I think it was, 4 to 2. Okay. In summary, this works and works in the genetically engineered mouse models. We do form T-cell memory that protects against rechallenge and can also cure remote uninjected tumors. In terms of making these retained agents, bigger is better and tighter binding is better. That's the take-home message of this. You want to be up to the left where it's most yellow because it's the biggest and highest affinity. In the canine sarcoma, we could safely administer these drugs, and we do get a biomarker response, which in humans is predictive of response to checkpoint blockade. And it's also safe and efficacious in oral melanoma. And in fact, Occam is interested in developing this as a product to be marketed in the veterinary market. The next step is into the clinic. And as I've said a few times here, both of these technologies are moving forward. And by the end of this year, we will have begun to see how this will work. So to return again to the people who've done the work, Noor started all of this work in my group, and she will now start as a faculty member at Penn in bioengineering next year. Yash Agarwal, a joint student with Daryl, did all of the alum development. Emmy did the type 1 interferon work and also the E3B binder engineering. Jordan and Ali have been spearheading the dog trial analyses. Daryl and I, all of the alum work is 50-50 on all of the alum work together intellectually. Tim Fan is the veterinary collaborator. And with that, I'd be glad to answer questions.
Okay, I think I'll start us off with a really quick one. Steve kind of always added this mouse serum albumin or canine serum albumin protein. Was one of the main considerations for that just to increase the size, or was also quite important?
Yeah, I'd like to say we did it for the right reason. We did it for the wrong reason. We put it in there as a buffer because we thought we don't want to be so nestled down inside of collagen fibril that it can't reach the receptor. So it was only intended to be a spacer. Great, off we went, did everything. And then only once we backed up did we find out we were lucky. Made it big enough to not leak out fast. Now this thing about retention once it leaves, that's actually bad.
Yeah.
So actually of the stuff that leaks out, it's now going to give you more exposure. So that's not great. So actually a company like Werewolf that does masking, they build their things intentionally so that when it becomes unmasked, it's also decoupled from the lifetime extension component. So what's left in the tumor is so small that if it does leak out, it gets cleared fast, which is smart. But we were just like,
Great, thanks.
As one of the mold of Langer disciples. So I guess I didn't understand the difference between anchored or binding that lasts for a day or 2 weeks and polymeric or any other controlled release system that lasts for a day or 2 weeks.
the accessibility, I suppose. If the depot is large enough that cells don't penetrate it, you'll have inaccessible, non-bioavailable material. So the intention, at least in the design here, is that from the moment of injection, it's bioavailable. That's the distinction. Now, on the surface of a depot, I'm sure there's also bioavailable material, but it's so we're not intentionally putting stuff where you can't see it.
So an anchored cytokine is as effective as a soluble cytokine.
Strangely enough, it's very strange, and I have to admit I don't understand it. When we put it on alum, the dose response is maybe shifted rightward twofold. I thought it would be much different, but that's a very robust result. I've got a student and I will come right back.
Okay, great talk. So I had a question. I've got 2 questions, but I think hopefully they're pretty short. The first one is, do you know the extent to which you can control how the collagen binding domain is accessible? So if you don't know where a tumor is, could you administer something systemically and then have some either cleavage event that you can activate or that the tumor mediates to trap your cytokine at?
Good. Now, at this point, I should give credit to somebody else's work. Jeff Hubble is looking at collagen binding domains that he doesn't mask, but he is also then masking the cytokine. So the reason I'm leery about systemic administration of a collagen binding protein is an awful lot of that ends up in the kidney and the liver, and it's just exactly where you don't want it. And so that worries me. But conceivably, you could mask both the collagen binding part and the cytokine part.
And my second question is, do you know the extent to which cells can over time start to internalize the entire construct? So do you not lose stuff because it diffuses away, but because cells just take it up and trap it?
And Noor built that into the model, the TMDD, target mediated drug disposition. In the collagen case, I think it always goes to the target cell because that's a pretty weak interaction with the collagen, tens to hundreds of nanomolar. The alum case, I don't think it does. I think it does. Maybe it patches onto the cell and stays there for a while. We have not done as many mechanistic studies on that as we probably should. The thing about something working is you just keep pushing it, working and doing all that stuff. And you say, gee, I wonder what they got. Should probably know that by now, but I don't.
You always ask the how or the why later.
Yeah, it's fun studying things that work. So
Thank you.
I want to follow up on Mark's question. So what are the kind of the dosing differences between a depot versus this anchoring approach? And then kind of on top of that, are there unique or what are the difference, kind of translational hurdles or regulatory hurdles you're approaching or encountering with the local regional treatment regimen?
yeah, so I suppose the differences in dosing are that with a depot release, with a release approach, you would want to be worried about that release exceeding consumption such that you would get systemic exposure. So that's a number of things that would be highly variable. So I would think that the safety margin you would have to build in would be fairly reasonable so that there's not a moment where suddenly you get a burst release of way too much cytokine. So I don't know. Now, what are the regulatory hurdles? We've had the pre-IND meetings in both cases. It's all green lights. We've done the NHP experiments in both cases. The alum, they barely know they got the shot. With the collagen, there is an MTD. But those two molecules are different too. Cullinans is an IL-2, IL-12 fusion with albumin, and Occam's is just IL-12. So they're very different there. I hope they both work. But yeah, so far there were no special concerns raised.
I'm just curious about, how do you think about like redosing and the frequency? Like, are you looking for certain markers, or is there a risk where you redose too frequently and, exhaust the cells, or?
Yeah, there are practicalities here that, supersede science. So one is, if you're going to combine with Pembro, they want you to do it on their schedule, which is every 3 weeks. So we're going to do it every 3 weeks. The other is return and compliance is an issue. So with some of the, TLR agonist trials, they would do 3 shots a week, multiple weeks. And even for superficial tumors, I think even somebody dying of cancer apparently gets to a point where they're like, this is just too much work. But we also want to go into visceral tumors. We want to do endoscopic and laparoscopic delivery. That's just not something you can do 3 times a week. And so that's a practical issue of just how often you should be able to do that. And I would hope and think that, something like the column that stays there for a long time should allow you to space that out.
Few quick online questions. For the canine experiments, did you use canine cytokines or the?
Yeah, cytokine, canine, collagen binding, canine albumin.
That was from Jeff Gang and from Eric Woods. Are both models, the sarcoma and melanoma, immunologically hot?
Yeah, the soft tissue sarcoma was pretty cold to start with. We have not done the nanostring on melanomas yet.
One last question.
Yeah, this is one. These responses could be fantastic for brain tumors, and local delivery could also be. Systemic delivery is a challenge, and maybe local delivery is one of the opportunities here. Have you tried anything in this direction?
There is a preprint on bioRxiv. I have a co-advised student with Polina Anikeva who defended his thesis yesterday at 9:30. Anthony Tibet. And he did that. He put it into B16s intracranially and also GLT61s, and got cures and delays. So it's interesting, but those are really hard experiments. That is interesting.
Okay, let's thank our speaker again.